Hello, everyone, and welcome. We'll be giving it a few seconds for everyone to join the webinar, and then we'll start. Okay, I think we are good to start. Hello again, and welcome to the webinar. My name is Chista, and I'm from the GRA Secretariat. I just go through a few housekeeping points, and then I hand over to Elsa for the rest of the webinar. So this webinar is being recorded, and after the webinar is finished, we will share this recording with those who've registered and also on the GRA social media. You can, at any point during the webinar, you can put your questions in the Q&A function, and at the end of the session, there will be a Q&A session where the questions will be asked. And that's it. With that, I hand over to Elsa.
Hi, and good morning, good day, and good evening to everyone. Thank you for joining this webinar from the BNI Flagship, and welcome. This is our first webinar in this flagship format. It's a little bit news to all of us, so bear with us. And since this is the first time we do this as the flagship, we will first start with a small presentation of the flagship. And if you have any questions regarding the flagship itself, it's a good point to, or a good time point to ask those questions after Cecile has been presenting. And after this little introduction to the flagship, we will hand over to Samuel for his presentation. Jacobo will introduce him a little bit more later. And yeah, so welcome. I want to start with introducing myself. So my name is Elsa Lagerqvist. I'm a postdoc at the Free University of Brussels in Belgium, and I work on BNI in Plantago lanceolata and how to incorporate this crop in cropping systems. Thank you. I think I will hand over to Cecile.
We might get introduced Jacobo first, and then I'll.
Yes.
Okay, so hello everyone. My name is Jacobo Arango, and I'm a plant biologist by training, and I'm the leader of the Tropical Forages program of the Alliance of Biodiversity and CIET. Thank you.
Thanks, Jacobo. Thanks, Elsa. And I'm the third co-lead of the BNI Flagship. My name is Cecile de Klein. I am a scientist with the Bioeconomy Science Institute in New Zealand, and I've been working on NH₄ emissions for my career, and have been in the last few years been focusing on BNI as an opportunity for reducing nitrous oxide emissions. And I will now go into a short presentation to explain a little bit more about the flagship and how we're hoping to, what we're hoping to achieve and how you could get involved. So I'll share my screen. Can you see this? Yep. Okay, yes. Great. So the GRA flagship project is about accelerating the development of BNI, biological nitrification and inhibition as a nitrous oxide mitigation strategies. And this has been developed in the last probably couple of years. We've been working on setting up the flagship. And because the flagship doesn't come with any funding, it is something that we have to do alongside our own work and our job. So it takes a little bit of time to get this all set up and working. But we're here now, and I'm really pleased that we have our first webinar under the flagship. just a little bit of an explanation on the background to BNI. I assume most of you will be familiar if you're joining this presentation on what BNI is. But biological nitrification inhibition it's been defined as a mechanism that plants can induce that reduces nitrification in the soil and therefore reduces the formation of nitrate and subsequently the formation of nitrous oxide. And we know from a lot of our work in the past that nitrification inhibition is a real has good potential for reducing nitrous oxide emissions. And now we're looking at, can we find a biological way to do that using plants as a medium? So there's quite a bit of work already been done on BNI in the past, and particularly focusing on tropical species. And it has shown a really good effect in terms of increasing nitrogen use efficiency. What we are focusing on here in the GRA flagship is really looking at it from a N2O perspective, reducing nitrous oxide emissions. So the reason why we are thinking a global collaborative approach is really needed is it seems to be that around the world the BNI research is rapidly evolving, and the research many research projects underway in many countries for various reasons, not only for N2O, but also for other reasons for nitrogen use efficiency. And it is a little bit of an emerging area, and there are not really good standardized methodologies available at the moment to really say, okay, these are the things you need to do to conclusively say this is a biological nitrification inhibition. So there is an opportunity to bring our knowledge together across the world and then really help standardize those methodologies, but also bring data together and see how we can collectively analyze those data. The work to date has been showing quite a lot of variability in the results. So being able to collate global data on BNI and how it affects nitrous oxide will really help to analyze the key factors that are determining or controlling BNI in pastoral systems, and also to see how we can develop it further as an N2O mitigation. So we have, as I said, the flagship was developed by a group of us over the last few years, and myself and Jacobo and Elsa are the co-leads. But we had a working group members that helped develop the flagship proposal. They're listed here, and really indebted to them, and want to acknowledge them here: William, Saman, Laura, Dave, Eduardo, and Rebecca. But in addition to that, we also had a seminar series that have been running for the last couple of years that really is a BNI expert group that has been meeting on a regular basis. And you can see some of the countries that are highlighted here where those BNI expert group members are located. So in terms of the anticipated outcomes or impacts of the flagship, we're really, as I said before, the flagship itself doesn't have any funding, so it's not able to do any actual research. But what we can do is help collectively connect up with others and connect up BNI research. And what we're trying to do is to really find out what work is going on across the world that we can come up with and join forces on data analysis. And we're using the G by E by M framework because it is a really neat framework to actually see where different people are focusing their efforts in. So the genetics, is about having plants that have the genetic background and the traits to induce BNI. Environment is really around what are the mechanisms, what is the conditions under which that trait is expressed by the plant. And management is really taking it out into the field and making sure that farmers can use it, and making sure that we understand how to maximize the BNI capacity of the species. So what we're also hoping to do, as I said before, is have agreed and standardized methodologies and guidelines to collectively work on. And then also it is a real focus on capability development as well across the globe. So training of early career researchers and also bringing in developing countries and opportunities there. And ultimately, what we're hoping to do, of course, is to find opportunities for funding and for collaborative research projects together. So the opportunities for everybody to get involved is to join the BNI webinars that we're starting off today. So we're gratefully accepting any offers to present from you all. So be in touch if you have anything. We're looking at to see whether we can either develop training opportunities or workshop on BNI methodologies or connect in with others that are doing this and advertise it across the network. As I said before, we're really keen on connecting all the research projects that are going on and the researchers, and that can be through webinars or through a framework or a database, an overview of BNI research projects across the world that we then can delve into. There is an idea of starting to look at a literature review or meta-analysis and or a methodology paper that we're trying to write. So that is also an opportunity to get involved with. And the GRA is particularly interested in low-cost entry for developing countries, and these are here listed as well. So there is opportunity potentially for research exchanges, bringing people to a BNI sources to a lab, to look at PhD or postdoc scholarships, and also exchanges or summer schools. So really, in summary, if you're interested to be involved or get more actively involved, please reach out. If you're keen to present at our webinar series in the future, if you have projects that you're working in the BNI space at any level, at the G by E by M framework, please let us know and we can collate it into our global overview of BNI research projects. If you have good ideas around methodologies or approaching, or you're using something new, or you have questions, feel free to share. Or if you are running a training program or have a conference or a webinar through that you'd like to advertise, again, we can do that through this BNI Flagship. And I've just listed the co-leads of the project again with our email addresses, and also note that we are working on a web page to develop for the GRA website, and the that will in due course be posted on this web address that is listed here. So with that, I conclude and see if there's any questions that people may have. I don't see any questions. Elsa, anybody has a question? If not, we can pick it up later.
If attendees have a question and would like to unmute and ask a question, I think attendees are able to raise your hands and then I can allow those to speak. So feel free to raise your hands now if anybody has a comment or a question. I saw one raised hand that disappeared, so. Okay. I think Jamie has a question, so
Yes.
Jamie, feel free to unmute and.
Thank you very much. My name is Jaime Garzón from the University of Maine in the United States. I was wondering, there is a plan with this flagship group to do also like a newsletter or something that we can have access, I don't know, maybe every month, every quarter, just to know the advances of the group across the year?
Sorry, I couldn't quite follow at the start of your question. What were you asking?
Is to know if the group has like a plan or an idea maybe in the future to have a newsletter.
Or a
Just to
newsletter. Sorry, I missed the
yes,
word newsletter. Yeah, that's that's a really good idea. We haven't really talked about it, but I think it would be a good idea to do that. So yes, it would be another way of connecting up and having newsletter. Yeah,
that
good
would be great.
suggestion.
Thank
Thank
you.
you. Yeah. Okay, if there are no further questions, we can pick up some other questions at the end as well, but then I'll pass on to Jacobo now to introduce Samuel for the main presentation of today. Thank you.
Thank you, Cecile, for introducing the flagship again to the group. So now it's time to have our main topic of the meeting, that is the presentation of Samuel. Are you able to project the slide, Samuel? Please. Thank you, Samuel. So today, Samuel will be telling us about his research on key driving genetic environment and management factors that influence nitrous oxide mitigation in tropical and temperate crops. So as you will see, Samuel is associated to the Alliance of Biodiversity and SEAD located here in Cali, Colombia. But okay, so Samuel, yeah, let me introduce him. So Samuel is a biochemical engineer from a local university here in Colombia. Now he's also a master's student on research and biological science at the University of Aberdeen in the UK. He's currently a research associate at the Alliance of Biodiversity International and SEAD, specifically in the Tropical Forages Program. So he has been leading activities of the International Research Project on nitrous oxide mitigation in livestock systems through biological nitrification inhibition. That's what he's going to present today. He has experience in experimental design, field and laboratory essays, and systematic reviews. So he has skills in soil microbiology and molecular techniques, bacterial and fungal isolation, DNA extraction, PCR, electrophoresis, culture medium design. So he has a strong background in bioprocesses, fermentation, and biofertilizer development. He also has experience in data analysis and statistical modeling using R and biobliometric tools, He has also knowledge in greenhouse gases measurements, forage systems, and soil plant microbe interactions. And he has experience working with international databases and evidence synthesis tool. So again, the work that Samuel is going to present us, he has been the leader and he has received a tutorial or mentorship by a different group of people from Ag Research in New Zealand, from the University of Aberdeen, and also from us in the Alliance of Biodiversity and SEAD. So now the floor is yours, Samuel. Good luck.
Thank you so much, Jacobo, and also Cecile and Elsa. Good night, good morning, and good afternoon for everyone in this meeting. My name is Samuel Diaz, and as my team said, I'm going to give you a short introduction and also show the results that we got from our experiment in SEAD. So please let me try to show my screen in this presentation, and I will give you all the information about it. Thank you so much. Okay, so to start with this presentation, I would like to I would like that you could get to know my team. So firstly, we got here 4 of the 8 people that were working in this international collaboration project. In the first place, we find Cecile Declain, who is the principal scientist in the project, in charge of giving the guidance for the decision making about this study, focusing BNI and nitrous oxide mitigation. She belongs to AG research team. In the second place, we can find Jacobo Arango, that is my boss at the Alliance Biodiversity and CIET, and he is the organizational leader and also the one supervising the activities executing in CIET and also in this project. In the third place, we can find Leonie Ferreira Martins, that is our statistician that was in charge of doing the modeling of this data for the statistical analysis and the meta-analysis in RStudio, and also gave us the quantitative insights. He belongs to the Penn State University in the United States. And in the fourth place, you can find me, Samuel Diaz Vasquez. I was in charge of doing the systematic review, the collection of the data, and also to carry out some laboratory and field experiments to measure this BNI activity and mitigation of N2O. I also am a master student at the University of Aberdeen, located in United Kingdom. And in the second slide, you can find your members of the investigation group. We have Sam Bowitt and Brent Barrett, and they are also scientists at AG Research Group, and they were focusing give a critical review of our results and also help us to interpret these results at the end to see if they make sense or not. You can finally find Jaime Garzón, that belongs to the University of Maine, a professor there, and he will help us to make the data set improvement to get consistency in the data, also help us in the interpretation of results. And finally, you can find here Cécile Goubry-Rangin, that is my academic supervisor at the university, helping me to develop the experimental processes. And that will be all about the team, our contribution to this work. So in our next slide, I'm going to talk to you about the process that we use to carry out this meta-analysis. So we have to previously know that this is an international collaboration project that was created to answer the current gaps about knowledge in BNI mechanisms and how these genetic, environmental, and management factors and their interactions affect the nitrous oxide mitigation in tropical and temperate forests. So that's why our specific objectives were to first compare these factors that are related to the nitrous oxide mitigation with the information that we got from different articles. And secondly, we wanted to identify the ones that were related to the nitrous oxide reduction. In our other objective, we want to also develop a conceptual framework and improve the experimental methodologies that we find about the studying BNI activity mechanisms for the nitrous oxide mitigation. And finally, to develop experimental studies to prove that these factors that we found in the meta-analysis are effectively significant, while proving laboratory and field experiments or glasshouse experiments. So now the big question is, so what is a meta-analysis? Well, so you have to know that we have to understand that this is a combination of data from different studies, and this make us able to calculate the single effect size, that is like the significance or the magnitude or the strength of the relationship between this specific factor and our response variable, that in this case will be the percent of reduction in nitrous oxide. So the process that we have to follow for make this process is firstly to identify the research questions. What is the problem? So as we said before, the idea is that we are going to find the factors that help us to mitigate nitrous oxide in crops. So after that, we have to search for literature to find the main keywords and to also find the most relevant articles and information. To after that, make a extraction of data and also an analysis of this data to be able to show the results, and in case that we can see an opportunity for improvement, to run another meta-analysis or another analysis. And this is the process that we follow. In our next step, what we did was to make a short systematic review to identify the main keywords that allow us to get into important articles that were related to nitrous oxide mitigation and BNI activity. So as our objective was to find these articles, what we did was to select 41 keywords that were divided in 3 groups that are related to nitrous oxide. So as you can see in this graphic, in the first table from the left to the right, you can find that the first group is the target subjects responsible for nitrous oxide reduction. So that's why we include terms as BNI, biological nitrification inhibition, nitrification inhibition, and the nitrification inhibition, because in these processes, nitrous oxide is produced, and those are responsible for the mitigation. In the second group, we find terms related to produce this mitigation, in this case, crops. That's why we put the normal name of the crops, for example, forage or grass. And we also include scientific names that we found in literature of crops that have reported BNI activity. For example, Brachiaria decumbens, Lolium perenne, Panicum maximum, grasses or forages that we know that have BNI activity. Also the rice, oats, and other crops. And finally, in this group, we find our response variable of interest, that is the nitrous oxide, and we include terms like N₂O, nitrous oxide, nitrification product, fructetsurate, and that's all. With these terms, it's important to clarify that we create a specific code of search that we introduce into these 3 databases that were Web of Science, Scopus, and Dataman, to be able to find the articles for the data extraction. Okay, so for the selection of articles, we follow Prisma methodology. That is a standardized method useful to carry out systematic reviews and also meta-analysis. And this basically consists in 4 steps in which we make an identification of articles and screening based on some inclusion and exclusion criteria. And also we do the eligibility step and inclusion of the final articles. So to give you a summary of the inclusion and exclusion criteria that we used, you have to know that we include articles that were experimental articles in which they evaluate the usage of crops and emission of crops. We include articles that have information in English and Spanish. Also articles that were related to nitrous oxide measurement didn't include information of synthetic, and that includes all the statistical information to we can make the meta-analysis. For example, we require that they have information about the standard error and the standard deviation. With all of this, we got a total of 23 articles that were used for the data extraction. Okay, so now that we got the articles, we got the keywords, it's important to define what were the factors that we used for the meta-analysis. So we used a total of 42 articles, and what we did with them was to divide them in 3 groups also: genetic factors, environment, and management. And with all these groups, we select the information about the numerical and categorical factors that we include from the 23 articles selected per treatments. So in the first group, you find 7 factors, like, for example, genes, plant age, plant genus, plant species. In the second group, you find factors like soil pH, soil nitrates and ammonium concentration, soil bulk density, and for example, water field pore space. And finally, in the group of management factors, we include the factors that talk about the treatments that the soil and the crops are receiving. For example, grassing or liming and fertilizer rate, and also information about the fertilizer type applied. So now it's important to talk about the response variable that we use to explain the nitrous oxide reduction and also the mechanism of BNI. As we said before, we are going to talk about the nitrous oxide reduction, but first it's important to calculate this response variable that is called the expected nitrous oxide measurements. And this is related to the fluxes that can be produced or the value of the fluxes based on the nitrogen fertilizer input and the emission factors. So this was calculated using the IPCC methodology that consists about the emissions reported of nitrous oxide and other greenhouse gases per country. And with this multiplication of the fertilizer nitrogen input and the emission factor, we get the expected nitrous oxide emission. You can also find the units here, and the fertilizer input was considered in kilograms of nitrogen applied per hectare, and the emission factor is in the unit of kilograms of nitrous oxide, of nitrogen in nitrous oxide per kilogram of nitrogen applied. Now, with this response variable calculated, what we want to do is to use the reported or the measured nitrous oxide value in the articles and compare this value with the calculated expected emission to calculate the reduction. And while doing this comparison, we can calculate the percent of reduction because we divide them between the expected emission. And you have to know or notice that we calculate the percent of reduction because in many articles, there was not too much information about BNI activity. And as we want to correlate this BNI activity with nitrous oxide mitigation, we select this response variable that was available in all the articles. And the units are the same, so it can be compared and calculate the percent of reduction. And now I want to also show the other equations that we use for the calculation of nitrous oxide. As we want to make a statistical analysis, it's important to calculate the variance and also the standard error. Because remember, we have a new response variable. We got from articles the information about the nitrous oxide measurements, but now we have this new response variable that is the percent of reduction. So we have to calculate the variance for the percent of reduction to after that calculate the standard error for the percent of reduction in nitrous oxide. So the first step is to take the standard error value reported in the treatments for the measurement values of nitrous oxide. And with this value, we can calculate the variance of the measured value. With this value, we use it to calculate the variance of the reduction, calculate using the expected nitrous oxide emission calculated, and multiply this per the variance of the measured nitrous oxide value previously calculated. And with this, we're able to calculate the standard error. And in case that we want to use a standard deviation, we can also do it using the standard error reduction, multiplying this for the n, that is the sample number in the experiments. Okay, that was related to the first part. And now it's important to clarify how the statistical analysis was done in our study. So you have to remember that our observations corresponds to the mean treatment. It means the percent of reduction of nitrous oxide that has the values of the standard error, and the standard error is named as a standard error of the mean. It will be called as SEM, reported in many articles of meta-analysis. Also, it's important to indicate that the approach used was the linear mixed effect models. And this is the right approach for us because it includes, for example, like in this equation, in which way corresponds to the response variable. This model helps us to include information about the fit's effect, the fit's intercept, also the random effect given per the specific study variability, and the residual error per every treatment. And that is related to the last comment that I was going to do, in which we say that the studies, for example, the study 2D and the study 14A and the other 23 studies, were included as a random effect to be able to explain the study variability in the models. And in the graphic below, you can see that we have the common approach applied, that is the fit's effect model, and the method or the method that we use, that is the random effect model, in which we see that every point corresponds to the treatment considered for the model. And we not only see the effect size plus the random error, we also consider the study specific random effects, shown in yellow in the graphics. So in the next part, we are going to continue with more comments about the statistical analysis. Also, it's important to let you know that the standard error of the mean was used as the opposite value. I mean, 1 divided the standard error of the mean, because it helps to give more precise estimates to the values that have greater influence in the model, and less precise estimates to contribute to the model. And also we use the approach of meta-regression because it helps us to understand the individual effects of every single factor considered. I mean, every single of the 42 factors considered over the response variable. That's because some environmental factors and some factors that affect plants and the environment are correlated. If we see it and we make an individual evaluation of there, it will be a clearer interpretation of the effect sites over the response variable. And also it's important to clarify that we use two types of R-squared. The marginal R-squared, that is the one that represents the proportion of the variance explained by the fixed effects that are the environmental and the management variables. And also we use the conditional R-squared, that is the one that represents the proportion of the variance explained by the entire model. And finally, we want to clarify here that we use 4 packages of RStudio to make the plotting of the graphs, to make the linear models, and also to visualize the graphics after the analysis. And now we are in the part of the results. It's important to notice that from all the factors that were selected, we find that only 12 of the, in this specific numerical predictors, were considered as significant, statistical significant over the % of reduction of nitrous oxide. So first I want to explain the table. We find a table of 4 terms in which we find the factors, the mean, or that I already told you that explain the effects of this factor over the % of reduction, that is our response variable. And if you find a bigger value, it means that it's more significant over the response variable. The standard error of the mean, that is SEM, and the p-value that tell us about the statistical significance if the value is lower than 0.05. You can see that we highlight that the available phosphorus concentration in this analysis was shown as one of the most significant factors due to its effect size of 182.1. And we also highlight that these factors, like the fertilizer rate and the soil ammonium concentration, were also significant over this response variable. That's why in our next stage, and that was the experimental field experiment, we use the fertilizer rate and the ammonium concentration in the soil as factors to evaluate the BNI activity. Now, in the group of categorical predictors, factors considered, we find the same type of table, but we also clarify the labels that we consider here. I only want to highlight some of them. For example, factors like the soil tester, and for example, the clay percent in the soil show a bigger effect size or mean that affects the percent of reduction of nitrous oxide. And also this label was selected for our experimental stage of comproval because the soils that we use to make these experiments are high in clay concentration. And finally, on the right side, you can see that the plant genus, for example, Brachiaria, and the species Brachiarium idicola show the higher effect mitigation, being coherent to the information that we can find in literature that these tropical forages can mitigate the nitrous oxide emission and mitigate the nitrification processes through texturation and through biological nitrification activity. In the discussion, I just wanted to show, because I guess that we don't have enough time for it, some articles that showed some relevant results that explain that our results are fine too, and that makes sense. I mean, about the availability of phosphorus and the soil depth, the nutrient dynamics. Some authors have found that, for example, when you use fertilizer that have more ammonium versus another fertilizers, it can help to improve or to improve or help that these crops can show BNI activity. Also, physical properties can help, like the soil tester, and plant species like Brachiaria are also reported to have BNI activity, higher BNI activity in crops and mitigate nitrous oxide. Finally, we just want to make some acknowledge to the Ministry of Primary Industries of New Zealand that was our sponsor for this project and that also covered my master's studies at the University of Aberdeen. That's possible because of them. And also the consultative group of international agricultural research, that is the CGR, group in which the Alliance of Biodiversity and SEAT belongs, and that is focusing agricultural greenhouse gases mitigation. And thanks a lot for your time. If
Thanks
you have
a lot,
some
Samuel.
questions.
Yeah, thanks a lot, Samuel, for the nice presentation. I don't know if there are questions from the audience. So I'm sure Samuel will be happy to take some of your comments or questions.
There is a question in the Q&A, Jacobo.
Is that on the chat or where is it?
No, the Q&A.
Okay, yes, now I see it.
Oh, yeah.
So, okay, so David from Ag Emission Center, maybe have a new name now, I don't know. In table 1, only 3 variables were highlighted, but there are other variables that also had large effects and were significant. Could you explain the rationale for selecting only the few variables highlighted in grid boxes?
Okay, perfect, David. I just highlight some of them just because of the time of the presentation. But I mean, in this case, we select the reason for selecting the first one. There is the available phosphorus concentration was because of the effect size. the size or the value or the magnitude of the effect size. Because as I described before, if you get a bigger value and also a p-value that is lowered to 0.05, it means that this factor is more significant than the other ones. And within the information found in the cycles, it seems to be like the most effective factor to influence the mitigation of nitrous oxide in these tropical and temperate crops. But also the other factors are also important because, for example, we have found that clay percent in soils can help us to mitigate soil deprecipitation. We also carry out some field experiments, and we found that when there was more precipitation, for example, we got higher values in nitrous oxide and soil depth also. But we highlight some of them because we decided, as we have to carry out another stage, another experimental stage on the field, we had to select just two of them. That's why I just highlight two for the explanation. But yeah, indeed, the other ones are important to the mitigation to be considered.
Thanks a lot, Samuel. There are some more questions here from Tony van der Verden. Hi, Samuel. Have you investigated interactions between variables?
Okay. Thanks, Tony. What we did to study their interaction, this is a really good question, was we studied the interaction more in the experimental part of the field experiments. Sorry, I just lost the shot. We carry out an ANOVA analysis after that, in which we made a comparison of the factors considered. And we found that in some of these experiments, the interaction between the factor selected, that was the fertilizer rate, and the crop with BNI activity that were from Brachyria sp species was considered, but we did it in the experimental part. I don't know if that answered the question, Tony. Yeah, because in the meta-analysis, as I said before, we only made the evaluation of the single factor versus the response variable, because some of the factors are correlated, so it's better to evaluate it individually and not in the same group. But that will be all.
Thank you, Samuel. So there is a third question. Why was the nitrification inhibition also selected along with BNI when data was collected for the meta-analysis? What could be a possible biological significance?
Okay, when we were doing, or when we were taking the, sorry, I will go back to that. Here. When we select the keywords to find the articles, we select the nitrification inhibition because we want to target or to find articles that were in which nitrous oxide measurements were reported. We found that in many articles that were talking about nitrous oxide measurements, we found the nitrification and nitrification involved. As you also know, the nitrification is an additional process in the nitrogen cycle that also allows us to get nitrous oxide. That's why we included in the first terms or first keywords that we use for the code of source that we use in these three databases. And yeah, what kind of... sorry, I was looking at another question. Yeah, that will be all the answer for that question. And the other question of Savarna, what kind of p-values were used? Any correction tests like Fisher or... yeah, Savarna. We use the 2K, no, Just let me go to the slide to see if I got this. And I do not remember this answer at the moment, but feel free, please, to send us an email so we can ask specifically to our statistician about this. Thank you. But we can keep in touch to answer this question to you.
Yeah, thank you, Samuel. Maybe another question in your meta-analysis. Only 23 out of 400 publications were used. Are you planning to generate and publish guidelines from the insights of your analysis so that the key measurements are made when researchers design future studies? Also from David.
Okay, sorry. Let me try to get into the question again. Okay, only 23 of the applications were used. Are you planning to generate and publish guidelines from the size of your analysis? Okay, we are currently working on a paper with these results, so we can show the results about the factors that are more relevant in the mitigation of nitrous oxide. But we are also going to share this data set. We are also in the process with the AG research team to do that. Yeah, but indeed, it can help to share our comments about it.
Thanks, Samuel. So there is a question from Pablo Lacerda. So thanks for your presentation. How phosphorus availability affect BNI?
Okay, thank you so much, Pablo. We were reading articles about this, and what is that? Oh, yeah. And we found, for example, this article that was a study that was done in peatlands, in which they were evaluating the effects of the application of phosphorus alone, nitrogen fertilizer alone, and the mixture of these fertilizers. For example, in this investigation, they found that when you applied phosphorus, nitrogen, and you also applied another nitrogen fertilizer, it can affect the emission of nitrous oxide. I guess that it's maybe because this, as this compound or nutrient is also important for the plant, it can help with the combination of our fertilizer, it can increase or decrease the emission of the cropland. But yeah, that's what I found on the results. We are also working on this for the publication, so we still need to get more time to explain big part of these results, but this is what I guess at the moment about this.
Well, thanks a lot, Samuel. I think we are almost at the limit of time. I don't know if there is any last comment or question from Samuel. If not, please join me congratulating Samuel and the team for the good work. And again, please get in touch with us if you have any suggestion or ideas for the flagship on BNI. Back to you, Cecile.
Thank you, Jacobo, and thank you, Samuel, for a really great overview of the work done. Lots of interesting results and lots of things to still consider, and good suggestions by some of the people that attended to think about for inclusion in the paper. So great work. So I think I'll pass on to Elsa. I think we have just a roundup to do now, and then we'll say goodbye to everybody. So Elsa, over to you.
Yeah, thank you, Samuel, for this presentation. Very interesting to see what you have been working on and your preliminary results. Yeah, I'm just gonna round up as we will have coming seminars as well. Maybe actually, Cecile, you are the one with the best overview. I don't know if you have anything in the pipeline yet. No? Then if
No, not
anyone
at the
is interested,
moment, no.
no. If anyone is interested in sharing something with the community, please let us know. We intend to keep these seminars on a reoccurring basis, maybe every second or third month or so. And yeah, if you're interested in joining the flagship in any way, also just let us know. It's really open for inputs and contributions and ideas. Yeah, I think that was all we had for today. Thank you everyone for attending.
Thank you, everybody. Goodbye.
Thank you. Bye-bye.
Thank you.
Bye-bye.
Thank you for joining. Bye-bye.